Okay, so yeah, we just want to run through a few things of what's going on, and I'll take you through a specific situation with some wheat powdery mildew issues we had there last year. So certainly when we talk about diseases, cereal diseases, but the same for all, we talk about integrated disease management. So everyone knows the disease triangle about, to get infection in a crop, you've got a number of factors. So host susceptibility, you need the pathogen there, and then the environment's the big player. And that's really where we're sitting in this current 221 season, certainly being wet early on and it's favoured a lot of diseases, but with a run of dry weather at the moment, we're really seeing some of that drop off. So we're sitting on a bit of a precipice about, do we need to manage some of this stuff or not? Because certainly those dry conditions is really, I'd say it's a free fungicide. It just takes the pressure right out of the crop. I guess the thing that Andrew Milgate down south come up with is it's actually more of a pyramid. So there is things as a grower or advisor that you can do to interact with these and limit some of these. So certainly some of the things we're doing in our systems can change, inoculum. So our rotation certainly defines what risk we're in, but also some of the things we're doing with stubble height and stripper fronts and leaving things higher, I think are going to create issues going forward with some of our necrotrophic diseases of leaving those spores higher up in the stubble. Certainly environment, there's not a lot we can do with the exception of sowing time and risk of things, but certainly what we find is there's massive microclimate differences within canopies. And we're seeing that with this year with just not much wheat powdery mildew around this year compared to last year with, canopies down a bit. We're not pumped up as much with N, and really the conditions not favouring that. And then obviously you've got your resistance over this side. So there are things that we can do. So you can play a role in influencing this and influencing your risk of disease. So in terms of what's actually happened this year, so another wet year, it's certainly been quite busy. So 202 diagnostics and inquiries have come in through our system, GRDC funded project. The little orange circles are where we're getting inquiries and greens where we're getting physical samples come in for diagnosis, correct diagnosis. Certainly into that southern Queensland area, which is also covered by Least or Snyman out of Warwick there. Certainly a lot more interest that year with a wetter year. So you guys were very dry last year, but this year, obviously being wetter, you've seen a lot more leaf disease around. So 24% of what's coming in has been yellow spot. So that is a big issue. There was a lot of wheat in the system last year to get stubble cover back. We certainly saw the fruiting structures maturing on stubble and put warnings out about that. And that's, really eventuated. So there is a lot of yellow spot, mainly in wheat and wheat situations. But there's also a lot of misdiagnosis of yellow spot continuing as well. Stripe rust has continued this year, so it was a big issue south of the Queensland border last year, and we learned our lessons on varieties like Bennett, which had dropped in resistance to that new pathotype, 198 pathotype of stripe rust, and certainly in that Queensland area, they're learning about that this year. Again, a lot of stuff is not disease related, so this is the importance of that diagnostic service getting a correct diagnosis. There's 20% of what's coming in is just physiological. Again, when we get that first run of frost or we get, abnormal frosts after plants have been growing nicely, they tip off, and just a reaction to that, and there is a few herbicide issues been around as well. Leaf rust has crept in wheat and barley, so certainly as you push up into that southern Queensland and just starting across the border, there's a few barley varieties in particular that are taking a bit of leaf rust. So you need to keep an eye on the more susceptible. So Compass is very, very susceptible. Also in Labrook, but yeah, you'll also get a susceptible rating in your Spartacus, some of the others. So one to keep a watching brief on. And we've got a bit of a watching brief on some stuff in wheats as well. Because we know some of those winter wheats, the Bennett, if you've got that in your region, very susceptible to the new pathotype of stripe rust, but also susceptible to leaf rust. So that's building up pressure within areas. Spot format blotch in barley, 11%. So it's certainly around mainly barley on barley, but we have got a number of situations where it's actually barley on wheat stubble. And we've seen this in our survey stuff. And Greg Platts, the barley pathologist, used to be with QDAF, used to talk about this. Is you can get saprophytic colonisation of senescing wheat late in the season if you've got moisture. And we're seeing that come through. We're getting wheat stubble out of these barley crops, and we're getting the Pyronophyteris, the net blotch fungus, directly sporulating on that wheat stubble. So that was a bit of a heads up with the wet finish last year. Fusarium crown rots around, and yeah, certainly as we move out into our more western environments, there's some quite severe infection levels there, given some of the cereal on cereal rotations they were forced into. We want it to stay wet so we don't get expression of that, but certainly big crops, big canopies, big water demand. We're not out of the wood on that. So And certainly a bit more net form and net blotch creeping in up this way than we've seen for a while too, but still at low levels. Okay, so that's a bit of a picture of what's around. Certainly when it comes to stripe rust, varietal resistance status has become complicated because now we've got multiple pathotypes circulating which differentially affect varieties. The beauty of that, big fan of the investment that GRDC's had over a long time and University of Sydney's put into the rust program, and their pathotyping stuff is really important. For me as a regional pathologist to get a heads up of what pathotypes out there, and we can put warnings about variety. This is from last year. This is the live maps. You can get the map on their site. They're mapping this year's stuff now. The yellow ones here are where we had that 198 pathotype. So it was the dominant pathotype that was around last year. So 67% of the samples that come in of stripe rust last year were actually that 198 pathotype. So this is an exotic introduction out of Europe or South America, which only come in 2018, but it's dominated since then. It's dropped some of those down. There is another pathotype, this YR239 pathotype in the red here. Had a bit up in the north, but was certainly a bigger issue down south and causes differential issues of taking out particular genes. Certainly the older pathotype, which come in Western Australia 2003 is around 25%, has got multiple mutations which have taken out other genes. One that's really interesting too is this 64 EOA minus one, which come out of Europe. This is one of the original ones from the first outbreak in 1979. It's still been picked up in that COBITI, University of Sydney survey at low frequencies, but we actually had issues with it up around Tamworth and just to the east of here too. It caused real issues in particular varieties. So this thing's crept around. It's got YR4 virulence. And yeah, this is just a picture here in our NVT trial where we got that particular pathotype, which is low frequency in the population, but it can cause differential reactions. So this is Catapult and Rockstar. What they look like to that particular pathotype, they're quite susceptible. And we've seen a bit of Rockstar pushed up into the north as an alternate variety up here, but this is happening commercially in paddocks in those little increase blocks. They're getting quite severe levels of stripe rust, so it's probably not a variety for us in the north. Okay, where's the best source of current ratings? The NVT site is the best site, I think. You can go there and generate the tables you want. There's an agreement from GRDC that they'll keep things most up to date here. Up the top corner here, disease rating tool. You can go in, select your state, select the varieties you want to compare. You can narrow that down to what you're interested in the actual disease resistances you want to look at. So quite a handy tool, and it'll spit that out very quickly. You can come up with a table pretty much like this, where you can see I've looked at leaf rust, the different pathotypes. This is original pathotypes of stripe rust here. The exotic introduction of the 198. And you can see it caused particular issues in some varieties. So we certainly saw Benefit was the one that dropped for a resistant reaction down to an SVS, and that really caused an issue. And being a long season winter wheat, that got the rust early, and then it creates pressure for our main season plantings with all those spores in the system. The other big warning on this one, if you're not across it, is that particular pathotype, which is dominating, means that all your Arroy type durums now drop to MS and need fungicide assistance. They haven't got adequate genetic resistance to keep them out of trouble here. As I said before there with Bennett, it's also going to create pressure with leaf rust. It's very susceptible to that as well. So as we've moved down into that central New South Wales, we've pretty much had to let Bennett go because we were spraying it at least twice for stripe rust, and once we got over that, because of the warmer temperatures that leaf rust likes, we just then had leaf rust come into, we were back into spraying it. So it was just too high maintenance. So they're rapidly looking for a replacement there. I'll probably get myself into trouble for this too. I've got a bit of a push on for northern growers. So north of Dubbo and that, don't get attracted by some of the southern wheat varieties that are high yielding in that environment, tend to not do as well up here. But when you look across their spread, they just don't have adequate resistance for some of our key diseases. I haven't got root lesion nematodes on here, but they're not good for those as well. So generally, as we push up, we've got a high dominance of chickpeas in our rotations. We can't afford to have high maintenance wheat varieties. We just logistically can't get across it in conducive years when we need to, manage ascochyta blight in, susceptible chickpea varieties like Kyabra. So might sound good, but you look at Lancer, it's got a solid package, and that's why it dominates the varieties up here. Until a replacement, high yield replacement with that level of resistance come, I really strongly urge to stick with Lancer. Makes my job a lot easier as a regional pathologist too. Okay, in terms of rust management, we've done these. These ones are gone, so we know it's a biotroph. Need that green bridge, volunteer wheat plants to survive. Varietal resistance, make sure you got the latest ratings. It really starts with your winter wheats. It's managing the pressure there. So they create the pressure early in the season, go on to your main season planting. So keeping that under control. Certainly upfront fungicides can delay it. So the flutriafol fertiliser is being used a lot further south in southern New South Wales and has been really effective. You know, and it's caused about a 70-day delay in the epidemic down there from what they saw last year in Bennett. So really effective at keeping it out. Big thing out too in those dual purpose ones, we tend to think that we can manage it with grazing. We just don't have the stock numbers. The only way to manage it with grazing is crash grazing, because you have to take the infected leaf out, open the canopy up in less conducive microclimate. When you haven't got the stock numbers, you get uneven grazing and the rust will keep kicking over those bits that aren't getting the grazing pressure. Once they get rusty and they're less palatable, there's no toxicity, but the stock just don't like eating it. And that's just a rapid reinfection point for that crop and surrounding crops. So yeah, not as effective as we'd hope. Certainly fungicides in varieties that haven't got adequate resistance, so if they're lower than an MR rating, then we certainly need to look at either 1 or 2 sprays in that. And it's all about, you know, protecting green leaf areas. So fungicides protect yield potential. They don't create yield themselves. So it's really matching that in. The other thing to remember is there is a real temperature interaction with this as well. So, you know, you need the moisture for infection, but then how quick it cycles and how quick you strip leaf with new pustules is temperature related. So stripe rust likes it where you add your max minimum together, divide by 2, closer to 15, that's stripe rust territory. And then leaf rust, the same process when it's closer to 18. So we tend to get stripe rust comes in early. Once it starts to go onto a longer cycle and seems to disappear, but it's just on a long cycle, that's when our leaf rust kicks in. So certainly a lot of it's starting to move into our leaf rust territory as we're warming now. So certainly vigilance there. In terms of genetic resistance, a lot of our varieties pretty much rely on adult plant resistance. It's a mechanism where the plant senses it's got the stripe rust infection within its leaf. Because it's a biotroph, it kills those cells and it kills the surrounding cells, and you strip out the infection. So it's got no means to survive. The issue with that is if you've got significant infection in your crop, and APR kicks in, you strip a heap of leaf doing it. So this is not how we want to use APR. This is why a combination of genetics and fungicides can actually stop this and protect green leaf area. How you actually want to use it is protection with fungicides until the APR gets expressed. And then what you might see if you hold a leaf up under high pressure, and this can happen outside of hotspots. If you hold a leaf up outside the hotspot, hold up to the sun, get some light through it, you'll see this flecking. That's the adult plant resistance expressing and taking out the infection. So that'll never establish an infection. So that's a good way to see that your APR has been expressed. But it's certainly an issue every year as to whether it's kicked in. So when's it actually become active? It very much varies with variety. So in our MR varieties, it kicks in around that first node stage, just 30, 32. MRMS, probably closer to 37, 39. Where our durums are, a lot of those are kicking in at that right at that autumn peak, or it can be a bit later. But certainly there's interactions with temperature, so lower temperatures can delay expression of some APR genes, and certainly nitrogen status can play a role. And when we're under high nitrogen status, varieties, particularly Suntop, plays up a bit and delays its expression of that adult plant resistance genes. If you're unsure and in those moderately susceptible varieties, certainly spraying at GS39 is the way to go. Okay, only trust it from full head emergence in that situation. So use the fungicide to overlap and protect. I'll move on to necrotrophic leaf diseases. Hopefully I'm going all right for time here. So certainly last year we saw a number of things around, similar happening here. A lot of spotting in barley is physiological, not related to disease, just related to stress. It's a bit of a wuss and produces all these brown spots, trying to pretend it's a disease. Spot form net blotch certainly was around, brown spots, yellow margins. So they're necrotrophs, they're killing cells and feeding on the dead cells. And the yellow comes from the toxin production that the fungus has that kills the cells. Net form and net blotch, a bit more last year and certainly creeping around. In the wheat, we didn't have yellow spot last year. That's certainly a different situation this year. So we needed that 2 consecutive years of wet weather is really favoured the fungus to get through its life cycle, produce those spores on the stubble. But there's still a lot of physiological, and we're seeing that again this year with tipping. This is in some kitty hawk. When it gets at frost, it tips its leaves, and yeah, it's not related to disease. So yes, correct diagnosis is important. But when it comes to necrotroph, first thing you do, okay, look at your spots, but try to find the inoculum source, which is the stubble, okay? So with yellow spot, it's wheat on wheat, and with your net blotches, it's barley on barley. You see these raised fruiting structures called pseudothecia, and that's where your primary infection comes from. Rub your finger across those, feel slightly abrasive. They've got these microscopic spines on the surface, and that's what you're rubbing off. Okay, and it may not just be that previous year's stubble. So here's some issues we had in 2019. Gary Onis took this. It was actually 216 stubble that met its moisture requirement through that drought, actually produced these spores on stubble. The 218 stubble in 2019 wasn't a problem for net blotch at all, because it just, fungus couldn't get it through its life cycle and produce these structures to reinfect. Okay, so certainly that drought conditions has restricted it, but now that we've got, infection levels last year, we had a wet summer, we've seen a lot of these fruiting structures on stubble, so the risk is there out in paddocks. I think Queensland's probably tracking one season behind, so there is low levels of yellow spots certainly up in Queensland, which is going to give you an infection this year, and a lot of infected stubble, which if it gets through its life cycle, you'll see these things on stubble. And you can see those, going into sign and know that you've got the risk and need to manage. So as I said, they're the primary infection. These guys swell up under moisture, release an ascospore, which gives limited spread. Then you get the leaf lesions, the lower canopy, continued moisture to get these bigger conidia produced, and they can blow bigger distances and can go between paddocks. But it's really a function of very wet years is yellow spot. You need successive rain events to keep the cycle going. Okay, certainly look at the maturity on your stubble, your 219 in particular and your 220. And for Queensland, you're going to be tracking 1 year behind. Okay, so when it's yellow spot, it's the most misdiagnosed disease in our northern region. You've got to tick a few boxes. So is there wheat stubble visible in the paddock? Can you actually see these fruiting structures on the stubble? The other thing is there's a clear distribution within the actual plant. There's bigger and more lesions on the lowest leaf, been infected for longer, more time for the fungus to expand, always got that tight yellow margin. Okay, the next leaf up will have fewer, they're not as big, fewer again and smaller. As they start, there's these brown spots, there's fewer of them, always got that tight yellow margin. It doesn't translocate the toxins. You can get a lot of general yellowing in the lower canopy, and we've seen a lot of waterlogging trying to get called yellow spot. It doesn't translocate the toxin and move it right through the plant, because then it gives everything else a chance to colonise that tissue. Okay, it's very selfish and only produces toxin in advance of where it wants to feed itself. Okay, the other thing to be aware of too, here's a lovely photo that just came in the other day, and got the samples put in. This is not yellow spot. It is a great mimic of yellow spot. The thing that was a bit of a giveaway here is we didn't have the variation distribution. They tend to be all big lesions in the canopy, and it all showed up quite quickly. You haven't got these smaller brown spots with a tight yellow margin at the top. What we actually figured out is zinc deficiency. So in this particular situation, he was marginal for zinc, and he actually used a herbicide called Logram, which we know exacerbates zinc deficiency. So yeah, very good mimic. So throwing a fungicide at this would have done nothing when it was never the yellow spot was never the cause. So certainly here to support. If you do get a correct diagnosis that you've got yellow spot, there is an app which you can download and can help you make those decisions of, given the resistance level, whether it's economic to spray or not. Last slide on this, and then we'll quickly talk wheat powder mildew. Just want to make the point. So whenever we talk fungicides, we tend to probably get tainted more by our experiences with rust, but there's a key difference. So I've used these fancy terms, biotroph and necrotroph, not trying to sound smart. Anyone who knows me knows that that's a big stretch. But rust being a biotroph means that they're keeping the cells alive and feeding within that system. So what we happen here is we get a slow build up in our epidemic. We spray here, we crash the infection because we can clean the infection out with the fungicide. We get a couple of weeks, 3 weeks of protection, then you get a slow cycle time again. Spray a susceptible right again, you crash the infection, 3 weeks protection, you take a while to get up. So it really fits with our growth stages. So that's what we say, a GS30 spray, so flag -2 leaves out, you crash the infection. The time it's only just starting to build up at tiny levels, that you can barely notice. GS39 spray, full flag leaf, you crash it again, and you'll get it through to full head emergence when the plant resistance gene. So this is using the genetics and the way that disease cycles to control it, even in quite susceptible varieties. The difference with your necrotroph, your yellow spot, your net blotch is they've got it quicker, so they've got a quicker cycle time. The other thing is you never clean the infection out. Because they're a necrotroph, they're killing the cells, and you've got no mechanism then to get fungicide into those lesions because they're no longer got activity in there. Okay, so you get a build-up, you spray here, you protect the green leaf that's there at the time, you stop those lesions elongating for that period of time, but once it wears off after 3 weeks, those lesions that are already there will keep elongating, and you're full-on production of inoculum if you've got moisture around. Again, it builds up, it never flattens, same thing, protection again, wears off, it's full-on show again, and you're always getting infected off the stubble. The stubble's an inoculum source throughout the season. So that's why we tend not to get as good a joy. So there's very little curative activity to days if you've got rose-coloured glasses on with your yellow spot with all your fungicides, whereas you can get 8 to 10 days curative activity on with your rust, depending on your cycle time. Okay, it's the difference with that one being clean cells and one keeping the cells alive. That's a fundamental difference. Certainly, is critical. So fungal diseases. So when we get a lot of this physiological, they concentrate towards leaf tips. Diseases don't do that. And you can see it's a specific tip here, which is a signature that happened at a set growth stage. That's my mobile phone number if you need to send pickies or samples, etc. And Liestel's mobiles who services Queensland. And just urging everyone, we're all in together. So the ability of you guys sending stuff in or letting us know what's going on is we can actually warn industry and have a bit of intelligence of what's going on. So submit those inquiry samples, and we can certainly alert you to things. Submitting samples to the rust pathotyping lab is critically important. Well, you get those maps of what the particular pathotypes. We've got a bit of a worry, which we'll hear about from Sydney Uni by the end of the week, hopefully. Bit of a concern about a certain leaf rust pathotype, and that's because growers and agronomists have been submitting samples. So it really gives us a heads up, and we can then, knowing what those particular pathotypes are, we know the spread of varieties out there. We can certainly then about some proactive communication as to how we're going to deal with it. And certainly Twitter can be a way of rapidly communicating. I'm on Twitter, and University of Sydney's their information certainly on Twitter as well. Time to quickly do wheat powdery mildew. This is just a bit of a plug to say that I guess we tend to think of fungicide resistance as being, a WA problem, or we hear about it in South Australia. Just to say that we're not immune from it in the northern region, okay? It certainly can be an issue. And I think the issue comes for us, partly we had issues with wheat powdery mildew. You can see the levels on the leaf and the head late in the season last year. A number of factors conspired for this level, okay? There's actually, after a run of drought years, we had a lot of mineralisation events. We were pumped up with N, which powdery mildew loves high N status. So we had really thick crops and just those repeated rainfall events. So disease that really loves humidity. But what we were worried about was there underlying issues with fungicide reduced sensitivity or resistance within that population. So we've got a collaboration going with Fran and Steve over at CCDM, who are the experts in this area. And certainly what we'll present there showed that there was some underlying issues. So the issue we've got here is they weren't necessarily in these crops trying to tackle them for wheat powdery mildew. They're actually trying to control stripe rust and leaf rust, and to some extent Streptoria down south in these. So the issue is when you're weak on resistance to those, you're locked into fungicides. So genetics is gone, and I think the next speaker will talk about the pyramid for fungicide resistance. You're taking out that genetic resistance. So no evidence of big issues with fungicide resistance in the rust, but it's the off targets when you're trying to control rust, and wheat powdery mildew being it, I think has become a bit of an issue why we need to rethink some of our strategies. So this is the survey, largely from down on the Victorian southern New South Wales border, up around Griffith, Hillston. Not all just high input irrigated, but largely was high input irrigated systems. And you can see the varieties, they tend to be Septa Vixen, very susceptible to wheat powdery mildew, SBS. So you've got no genetics backing up. What you're looking at here is the frequency of genetic mutations within that population from the samples that were sent in. I'll cover the quoll first. So quoll is the strobilurin resistance, as oxystrobin as an example. You can see here we've got some high frequencies in the populations, in northeastern Victoria and some of these other sites. So this is complete resistance when you get into that one. The other thing that we were amazed about was a gateway mutation for the triazoles or DMI. So this doesn't mean your triazoles are gone. It just means that you're potentially exposed. You need underlying mutations after that to actually have issues. So certainly, a frequency of that. And this means these guys are totally compromised. Whereas, with the gateway mutation, potentially reduced sensitivity to some of those actives. And the frequency really surprised us how much was actually there. So I don't think that it hasn't been happening. Certainly some of the agronomists have been seeing issues with wheat powdery mildew in those high input irrigated systems probably for the last 4 years. So it's more that we just hadn't looked and it wasn't a reality. So quickly on wheat powdery mildew, it is different species that gets into wheat and barley, so they don't jump across species. We don't know what's going on now with barley powdery mildew. Survives on the stubble is these Cleistothecia, the black structures. That's how it survives between seasons. Certainly, wheat on wheat's a higher risk for wheat powdery mildew, but they can blow distances. They've got a wind-borne conidia spread within crops and between crops. And certainly, if you've developed resistance within, fungicide resistance within your population, you can blow that into your neighbours and even further. So what one does can affect the others with this particular pathogen. This is an interesting one because it's a real function of high humidity. So humidity, you get powdery mildew infections at mild temperatures, 15 to 22. But once you really hit 85 and above, that's when it's really take off. So it'll go a bit above 70, but above 85. So that successive wet days is what really drives it, which happened last year. I've had basically one report of wheat powdery mildew this year from in Mitch in Queensland at the moment. So although we're wet, it's certainly not maintaining that humidity for as long. So it really hasn't kicked in quite yet. So hopefully we'll see it not eventuate this year. So I say pathologists, the best way to not see a disease is do trials on it, which is what's happened. So we probably see no wheat powdery mildew this year. Anyway, thicker canopies are certainly conducive, that conducive microclimate, and that leaf nitrate level as a food source also favours it as well. Sorry. So how to manage it? It's a bit of an incomplete picture, but certainly people are on top of it. So the issue with this is we know that if you've got a high frequency of that strobilurin mutation, then you can't use your strobilurins. But we have this widespread gateway mutation. So what that actually means, we don't really know at the moment. The issue with that when you talk to Fran is that there's actually up to 4 new underlying further mutations, and they haven't been described previously anywhere else. So if they had been described, we have a fair heads up of which of the actual actives are more compromised. So yeah, that's been an issue in slowing things. So they're into some glasshouse work at the moment. The only thing that really we've got to share at the moment is there's some detached leaf stuff and a lot of caveats around this that doesn't necessarily mean how it'll perform in the paddock. It's just a bit of an indication showing that your wild type isolates here, how much of the active you need to kill off, half of the fungus on the leaves, in attached leaves, compared to your mutants. So you can see here, certainly you can still control it. You just need more concentration of the actives to do it. Okay, so they can be still active, but that's what it is. So we think it's an overexpression of binding sites. So it means you need more of the active to take out than what you used to need to control. So for this best bets issue, certainly follow the AFRIN guidelines, which are going to come up in a sec. Rotate and mix modes of actions. We're a bit limited. If you've certainly lost your strobilurins with that core mutation, you're really only got your DMIs left. Okay, because your SDHIs aren't great mildewicides. There is a group 5 spiroxamine, which is only registered in barley, so it's an option in barley, but it's not registered in wheat. The other thing is, which I learned too, so I'm learning about fungicide resistance I go to, rotating the actual DMI actives, your actual triazole. So not using the same triazole consecutively, on your first spray to your second spray. There's certainly a way of limiting how that's going, and certainly monitor the efficacy. So it doesn't perform as you expect, then you've probably got reduced sensitivity issues. So going again with the same active is just not the smart way to go. Also consider what the concentration of that active is. So some of those, we saw some of the late sprays inadvertently with Prosaro, so prothioconazole plus tebuconazole. Bit of an indication that the tebuconazole might be doing the heavy lifting with wheat powdery mildew, which if you use that in a mix, then you've automatically halved your tebuconazole concentration in that product. So just wanted to be a bit aware about what's the actual active levels that you're putting in some of those mixtures. And I think we haven't got the evidence to say that we need. Normally we think of with resistance issues, we chuck out all the old triazoles. I think that's probably not the evidence to do that without further testing. So certainly we know ciproconazole, which is in like your Amistar Extra, is quite a good mildewicide as well. Folioflutriafol, and yeah, we've got questions on how good tebuconazole. I know the one case we had up north here that was sprayed with a full rate of tebuconazole late when it was getting decent levels of powdery mildew worked quite effectively. So that was a good feedback from the agronomists. And there is a trialling, which means that we won't see any wheat powdery mildew down on irrigated sector with FAR, Nick Poole's crew, and that's the agronomist James Murray's managed to find that plant there. And that was all I was going to talk about. Thank you.
Great, thank you, Steve. That's a lot of information to take in. Don't forget, everyone, if you've got any questions, feel free to pop them into the question and answer panel. We've also been joined by Fran Lopez Ruiz from CCDM in WA. So if you've got any really curly questions, he's happy to help out as well. So thank you. And I'll now hand over to Levy if you want to just share your slides, Levy, and go from there.
Thank you, Bridget. Hello, everyone. 1 second until this starts, and I hope it's here. Yes, so let me begin with these two leaflets that I saw the other day in a GP's office, because I think that the antibiotic resistance in medicine and the fungicide resistance in crop health are basically two sides of the same problem. And this is the emergence, a widespread, worldwide emergence of microbes, pathogenic microbes that do not respond to treatments with antimicrobial drugs. So in our case, fungi, crop pathogenic fungi that cause crop diseases don't respond to treatments with fungicides. And as one of these two leaflets say, it's time to take antibiotic resistance seriously. I think it's time to take fungicide resistance seriously. A simplified way of looking at the fungicide resistance development is shown in this figure. And this figure comes from the guide to the fungicide resistance management in Australian grain crops. This is the guide that Bridget mentioned in the introduction and even showed it to you. It can be downloaded from the EFRN website, and I would encourage everyone who is interested in managing fungicide resistance to look at this great resource. It was published this year by GRDC. It's a 50-page long guide, and I think it's a really high-level, amazing resource for everyone dealing with these problems. So in short, those blue dots on the leaf are the fungal pathogens that are sensitive to the fungicide that is applied here. And if there are some that are resistant, those will obviously take over and will be able to cause disease even after the fungicide application at label rates. So this resistance is associated with a number of DNA markers, and we have heard about these DNA markers in Steve's presentation, and I will also mention a few more later on. I really like this map of the fungicide resistance cases identified in Australian grain crops. This is an amazing resource coming from Center for Crop Disease Management, CCDM, Fran Lopez Ruiz's team. And this is the latest map, an updated map. As you can see, it was updated in July this year. And there are a number of, sadly, there are a number of spots there that were not there last year. I will come back to this in a minute. You can also see there a list of diseases that are affected by fungicide resistance in terms of management, at least in the regions where fungicide resistance was identified. And there are three categories there, as you can see, L, RS, and R next to each disease. Let's take the first one, barley powdery mildew. This L means that the fungicide resistance or reduced sensitivity was detected in the laboratory, and I will come back to this in a minute. RS means it's reduced sensitivity. Again, the next slide will explain what does this mean. And R is resistant, which means that there is a field failure with at least some of the fungicides. In this particular case, DMIs or azoles or triazoles group 3 mode of action fungicides. Some of them simply failed to control the disease in the regions where this was detected, not everywhere, just in those regions. So this map is also a wonderful resource if you want to be updated about fungicide resistance in Australia. And a little bit about the terminology that we have already used, Steve has already used earlier. When we say that fungal crop pathogenic population is sensitive to a fungicide, to a certain fungicide, it means that the fungicide works perfectly at label rates. Reduced sensitivity means that it still works in the field. However, higher rates may be more effective, and there is certainly a high risk of developing full resistance. Full resistance, which means that simply that kind of active is not working anymore. Field failures were detected, and the fungicide should not be used, at least in that region, to control that particular disease. To declare, to state that a fungicide has failed, so there is a resistant crop pathogenic population there, or to talk about reduced sensitivity, lab detection is always needed. So this has to be confirmed in the laboratory. Why is that? Because we know very well how many other circumstances may lead to failures of pesticide treatments, such as faulty products, poor application timing, operator errors, weather conditions, and so on and so on. So what are we doing in the laboratory? We try to detect if there is reduced sensitivity to that particular fungicide in the target fungal population. If these fungi, if these crop pathogenic fungi can be subcultured on agar media, so mostly they are necrotrophs, then these lab fungicide resistance tests are done in culture. And if these are obligate biotrophs, such as rusts, powdery mildews, downy mildews, then we need detached leaves to be used to detect reduced sensitivity in fungicide resistance. And as we have already mentioned a number of times, there are these DNA markers that can be detected in the laboratory, and those are associated with fungicide resistance or reduced sensitivity. Coming back to this map, one of the new cases is the case that Stephen talked about earlier. This is the strobilurin resistance in wheat powdery mildew in New South Wales. And there is another case that has been added to this map this year, actually. It's the resistance of Septoria triticide blotch to strobilurins again. And none of these are in Queensland. However, I think it's important to know about these cases. And just a really recent news from the EFRAN website. It was published there at the end of July. You see, this is the strobilurin resistance in Septoria triticide blotch populations. This was detected by Fran's team at CCDM as part of a routine annual monitoring of fungal crop pathogenic populations. And it was detected before field failures were actually noticed. So this year, CCDM's advice, and please correct me, Fran, if I'm wrong, is to monitor the efficacy of fungicides that include strobilurins or QOI fungicides. The mutation that was mentioned in Steve's presentation, the G143A mutation, confers complete resistance to all strobilurins. So azoxystrobin basically, and pyraclostrobin here in Australia, these are the two strobilurins I think that are registered. So once this mutation becomes widespread in this case in the Septoria triticide blotch populations, strobilurins have to be forgotten. The bad news is that in these Septoria triticide blotch populations, reduced sensitivity to a few old DMIs, such as tebuconazole has already been detected earlier. And you see these are the green spots here, quite widespread on this map. So these populations have already exhibited reduced sensitivity to a DMI fungicide. Now there is complete resistance to all strobilurins. The good news is that a few other DMIs, such as epoxiconazole and the SDHI fungicides, are still active against these pathogens. So there are still options to be applied, at least in the region where this strobilurin resistance was detected in South Australia. Now, coming back to Queensland, we only see a blue dot there, which is DMI resistance in barley powdery mildew populations, which was detected in the laboratory, then reduced sensitivity, and then also complete resistance was documented in that region. How do we know that there are no other cases in Queensland, no other fungicide resistance cases? There are two ways of doing that. Of course, the most important one is that a number of diverse fungal pathogen samples are being sent annually to CCDM, to France Fungicide Resistance Team, for regular monitoring. And we have also started a smaller pilot project here at University of Southern Queensland with the support of the Queensland Department of Agriculture and Fisheries. Our collaboration is known as the Broadacre Cropping Initiative, or BACI, and under this umbrella, we actually started to look at a number of high-risk pathogens in Queensland. We have done some work about the rice blast pathogens. I won't talk about this because rice is not a GRDC crop. And we have also focused on mung bean powdery mildew population, and more recently on net blotches of barley, which is in progress. All our work is done in collaboration with a friend's team at CCDM. So just a few words about mung bean powdery mildew. Mung bean itself is an important legume crop in our region, and also further north in Queensland and also northern New South Wales. The highest production, I think, was in 2016, when altogether 180,000 tons of mung beans were produced in this area. And up to 40% yield loss has been documented when no management strategies were in place and when the environmental conditions were conducive. Now, growers are really used to this powdery mildew, and they don't even consider the yield loss. The management consists of fungicide applications and not much more, although there are a few varieties that are a little bit more resistant to powdery mildew than others. There are two fungicides available. One is tebuconazole, and another product contains a strobilurin, azoxystrobin, and tebuconazole, a combination of these two fungicides. Field failures have been repeatedly observed when tebuconazole was applied at label rates and according to all the instructions, and repeated applications of tebuconazole. After these repeated applications, powdery mildew was still there and covered almost all the green areas of the crop, including the pods. And as I said, the yield loss is quite evident there. Now, the situation is more complicated because we have recently discovered that the disease is caused not by one powdery mildew species, but two. These actually represent two different genera, so they are really different powdery mildews. The symptoms are exactly the same. The first samples with this new powdery mildew species were collected by Lisa Kelly at DEF, and the whole work, which has been published earlier this year, was done in collaboration with Lisa and with Nilufar at USQ. We are also looking at a potential case of DMI resistance there. And as a quick and dirty approach, we have just sequenced those DNA fragments that may contain one of the well-known markers associated with DMI resistance in mung bean powdery mildew samples in both species, by the way, that cause mung bean powdery mildew in mung beans. And the mutation, this well-known mutation, was not detected, which is a good news. But very recently, we started a discussion with the friends group at CCDM, because it may happen that we are not looking at the right spot. We may not look at the right DNA marker there. So the saga continues. And just as an interesting fact, this year when we went out to check a paddock which was repeatedly sprayed with tebuconazole, and even after these repeated applications of the fungicide, the crop looked like on that photo made by Lisa, as you see, complete coverage of powdery mildew. We noticed that the mildew mycelium is not that white. It's a little bit brownish. So a quick microscopy revealed that actually the whole powdery mildew mycelium was heavily parasitized by an intracellular hyperparasitic fungus known as Ampelomyces. These hyperparasites penetrate the powdery mildew mycelia and eat up the powdery mildew cytoplasm, the powdery mildew materials internally, and then they happily sporulate there and spread across the paddock. So it was hard to believe that the whole paddock was, first of all, infected with powdery mildew, and later on, these hyperparasites, despite repeated application of tebuconazole, took over. And wherever we went, we drove around this quite large paddock, we found these hyperparasites in action there. We cannot rely, of course, on these hyperparasites, because first of all, they come late in the season when they may not contribute much to the powdery mildew disease control, but it was interesting to see them there in action. This was an irrigated mung bean paddock, by the way, which is important. Probably that's why the hyperparasites had chances to develop and become really widespread over there. This is just an interesting thing. These hyperparasites have been registered as biocontrol agents in Europe and North America, but not in Australia. So we cannot apply them to control the disease instead of chemical fungicides. And to wrap up the Queensland update, I would like to mention a two-day workshop held here at USQ in March this year, and orchestrated in an amazing way by John Cameron and Nick Poole. One of the exercises here at the workshop was the use of a powdery mildew app, a decision support system, which was developed with the support of DBIRD, of course, GRDC, and also Becky. This app is useful if powdery mildew is there in mung bean paddocks, and the growers want to estimate whether it's economic to spray once or twice, and what would be the yield after these fungicide treatments. So overall, I think this was a really useful workshop, and probably the very first one on fungicide resistance in Queensland. So to wrap up, it looks like in Queensland we don't have too many cases of fungicide resistance yet. At least these were not documented yet. So we are still in time to prevent the development of a number of fungicide resistance cases in a number of crops. And these are the guidelines, again, from the fungicide resistance side that is available on the EFRN website. The main message here is that the integrated disease management should not be based on fungicides. It should be based on the right choice of varieties, of course, then all the agronomy and stubble management and crop rotation and all the other key elements of integrated disease management, and then use fungicides if needed, and only apply strategically and mix the mode of actions and stay within labor rates. These are the main messages here. So I would like to stop here and thank you for your attention.
Fantastic. Thank you, Levi. That was great. I just wanted to quickly ask a couple of questions. Oh, sorry, I'm just going to share that. So just a reminder that if you have any questions, feel free to pop them in the Q&A. Otherwise, feel free to head to the AFREN website where there's a whole heap of information available. Levi, just a question on the mung bean powdery mildew. You've mentioned 2 fungicides that are registered for that disease, but only tebuconazole might have resistance issues. So I'm just wondering what's happening with that other fungicide that's registered for mung bean powdery mildew.
Yes, so this is a good question. And during this AFREN workshop held in March, we learned from the agronomists and growers who attended this workshop, over 20 participants altogether, that the other fungicide is also used to some extent by some growers, although it's much more expensive than tebuconazole alone. So, so Custodia, that's the other fungicide, can also be used. It is used by some growers, and it would be a good idea, I guess, and it would not be a big effort here in our lab or at CCDM to check if strobilurin resistance is there in the mung bean powdery mildew populations, just on the safe side. So the other fungicide is much more expensive, but it has been used, and maybe there is a management option there if tebuconazole fails. Thank you.
Great, thank you. We've got another question also about management, and the question is, has foliar application of micronutrients such as boron, etc., based on SAP test being considered or tested as part of the management? So I'm not sure who would like to tackle answering that one. Fran?
Was it a specific mung bean powdery mildew question, or do we know what it was?
That's a good question. It doesn't actually say.
Well, I guess in short, no. No, they haven't been considered. So I don't know. I guess if we could get more information on that question. So no, we are not testing for that. No.
Okay.
Yeah. So it's certainly well known that there is micronutrient actions with particular diseases can make them worse. So certainly in the cereals, we've got a bit of a heads up with having a few bacterial blight issues last year on the Liberal Plains, but certainly elsewhere, which seems to be linked with some manganese and zinc deficiency issues. And when you look at research papers, you find a link there with that things as well. So it's more that if the crop's struggling for one thing, it becomes a bit more exposed to infection as well. So, but I'm not sure. Yeah, so it's part of an integrated. So we don't want to get our deficiencies wrong, but yeah, can certainly help. It's not like we can control it purely with nutrition in susceptible varieties.
Great, thank you. I hope that answers that question. And I had one for you, Steve. You mentioned about rust and wheat powdery mildew being really regionally based, but that they can sort of move between regions. Is it the same with fungicide resistance?
Yeah, well, that's the potential for something with wheat powdery mildew where it can blow distances. Yeah, if you develop resistance within a population, in theory you can spread that you know, to other regions with that windborne phase. The other way I'm a bit concerned that some things can get around, you know, probably more with Netform and Netwatch, where there's been some issues, is seed. So things that are seed-borne, we could inadvertently, by moving seed between regions, potentially, well, not just move, you know, fungicide-resistant isolates on that seed, but also, I guess, more aggressive isolates too. So we certainly know with our chickpeas, there's that Curao isolates, which is worse for ascochyta. There's potential, if we're not on top of our game, that we could move, more aggressive isolates as well, or fungus or resistance isolates between regions. So we basically give it a free ride in that situation. But yeah, certainly with the powdery mildews, it can blow distances, so that can be an issue. It's a bit more of a social side to it. Fran might like to buy into.
And adding on to what Stephen has been talking about, moving hay is also, potentially quite problematic, right? So we don't know what we are moving around with. If you're moving hay that could be infected, and that infection could actually, contain some resistance as well. And so we are basically contributing to the spread of resistance. So the movement of the pathogen is something that, unfortunately, we cannot restrict. So the only way that we can control that is by, working as Stephen was in one of his slides, as a community. So we are all in this together. So we need to make sure that all of us, are just playing a role here and are just doing the right thing.
Yeah. Quickly, Marcia's brought back into, I think it is very much part of programs in that agronomists are managing for disease, but also nutrition. So I know a lot of work gets done, particularly in my region, on micronutrients, and certainly if you look in a lot of that West Australian stuff, they talk a lot about potassium and spotful net blotch, the interactions there. So it is something that is not dismissed. It's but it's just part of good management of crops. Is they're certainly looking at what they're taking off in removing from paddocks, in grain, or if they're cutting hay and trying to replace those things. So trying to get on top of deficiencies, which there's other whole investments in that area within GRDC, which we certainly, as a pathologist, keep across where, what are the likely deficiencies in soils in our region. So yeah, we certainly don't want to get out of balance. So try to grow healthiest crop as we can, and then manage that as well, the diseases as well.
Great, thank you. We've also had another question through around if there's something to spray on stubble to stop spores from the previous season. Mancozeb has been suggested. Is that something that's recommended?
Diesel. And then hit it with a match. So quick one on that. This is a lovely one. You can't spray dead tissue stubble because there's no mechanism for uptake. So all your fungicides are water soluble, and they actually move through the plant from where they hit it. So if you're looking at a foliar application, from where they hit a leaf, they can only move in one direction, to the leaf tip, because they all move within the xylem, the water flow tissue. Okay, so we can't spray stubble and get rid of inoculum or spores within that stubble from forming. There's just no mechanism for uptake into stubble. So, mate, I can tell you, plenty have tried it, and not a lot of joy. Well, crown rot's the one I keep getting asked about all the time, and no, it does not have an effect because there's no mechanism for it to get into the actual stubble.